Okay, so Assalamu alaikum everyone. My name is Rawad Turko, and today, inshallah, I'll be going over plasma lipoproteins 1 and 2. So I understand that this may seem very last minute to a lot of you guys. Of course, we have our MOL midterm tomorrow, but I just thought I'd go over the slides and just go over any concepts that we thought that seemed a lot easier than they actually were. So this won't take long, inshallah. They're very easy lectures. Hopefully, you don't have that much trouble with them. So let's get started. Now, as of the slide, there's not much that we could take away. We don't have to remember any of the structures, any of the rings, the carbons, none of that. All we have to remember from the slide is that once we attach a fatty acid group to our third carbon on the first ring of the cholesterol, we get a cholesterol ester. And this is important for later when we talk about lipoproteins, because cholesterol esters are actually much, much more hydrophobic than cholesterol. And that's why when we look at the lipoproteins, they're buried deep into the core. So what I want to do for this slide is I want to correlate one lipoprotein to each of these methods of lipid transport. So for the first one, we have transport of fatty acids from adipose tissues to other tissues. Now, the key word is fatty acids, because once they notice that they said fatty acids and not lipids or cholesterol or any of these things, this tells us that these are free fatty acids. And what we know about free fatty acids is that they're transported through albumin. So next we have transport of dietary lipids. Dietary is another keyword, from the intestine to other tissues. Now, this tells us immediately that we're talking about chylomicrons. Transport of endogenously synthesized lipids from the liver to other tissues. This is VLDLs. And finally, reverse transport of cholesterol from extrahepatic tissues to the liver. This is HDLs. So what are plasma lipoproteins? So plasma lipoproteins are just another method for us to transport lipids and cholesterol throughout the body. Okay, so they're made up of phospholipids and unesterified cholesterol in the surface membrane. This is because these are less hydrophobic. Then we have cholesterol esters buried deep in the core, because as I mentioned, cholesterol esters are much, much, much more hydrophobic than unesterified cholesterol. And finally, we have apolipoproteins. Now, apolipoproteins is a very important concept and it's very high yield. So make sure you understand this concept very, very well, and we'll be going over it very shortly. So from this slide, there's also not much that we can take away. But what I want to do is, again, I want to attach an example to each of these things that are mentioned. They said most lipids and apolipoproteins can be transferred from one lipoprotein particle to another. So let's add an example. They said most lipids. So which lipoprotein do we know can transfer its own lipids to other lipoproteins? Well, that's HDL. HDL can, of course, transfer its lipids to LDL through CETP. Now, most lipids and apolipoproteins. Again, this is HDL, because HDL can donate its ApoEs and ApoCs to VLDLs and LDLs and all of these things. So what are plasma lipoproteins? As I mentioned, their function is to keep the components, their component lipids soluble in plasma. And they do that by packaging them and then transferring them through the bloodstream. They transfer lipids to and from the tissues. However, this transport system is not perfect, resulting in gradual deposition of lipid in tissues. Now, which lipoprotein do you guys think they're talking about here when they said gradual deposition of lipid in tissues? They're talking about LDLs. And LDLs, as we know, are bad lipoproteins. And they cause atherosclerosis. So you can see here that we have a normal artery, and then when we have this gradual deposition of lipids in the arteries, we get atherosclerosis, and this is narrowed arteries. And this of course, can cause cardiovascular issues, strokes, and all of these things. So they're composed of a neutral lipid core, triglyceride and cholesterol esters. That's because these are very, very hydrophobic, surrounded by a shell of apolipoproteins, phospholipids, and free cholesterol. Okay, so TAG and cholesterol carried by lipoproteins are obtained either from diet, exogenous source, and this is, of course, chylomicrons again, or de novo synthesis, and this is VLDLs. Again, for this slide, there's not much that we have to know. All that we have to remember is that the goal value of total cholesterol is less than 200 milligrams per deciliter. Okay, so if we have anything above 200 milligrams per deciliter, this would be hypercholesterolemia. Okay. Another thing I want to point to is that elevations of plasma triglyceride usually are caused by increased VLDLs. Now, this is very, very important, and this caused quite a bit of confusion. And I'll be coming back to this in a minute once we reach another slide. And elevations of cholesterol usually are caused by increased LDL. And this makes a lot of sense because when we have high LDLs, we get increased risk of atherosclerosis and all these things. And these things are caused by hypercholesterolemia. Okay, so after we eat, let's say we have a meal right here. This is when we have our meal, and it happens to be a very high lipid meal, very high lipid content. Okay, so after 3 to 6 hours after we've had this meal, we get transient hypercholesterolemia, and this is because of the chylomicrons. So the chylomicrons, we know, the chylomicrons are package exogenous sources of lipids. So they're mainly diet focused. Okay, that's when we have our meal, we get transient hypercholesterolemia. Now, this hypercholesterolemia is not indicative of any clinical cases of any diseases of any of these things. That's why when we go to get our blood cholesterol checked at the doctor's office, we're told to fast for at least 9 hours before we go in to do our blood test. And this is because if we eat right before our blood test, we might go in and we have hypercholesterolemia, and this might cause issues with the results, because this is not indicative of any diseases. This is just normally what happens right after we eat a meal. So when we talk about density of lipoproteins, we have chylomicrons, which are the lowest in density and largest in size. When we talk about density, we're actually talking about the protein aspect of the lipoproteins. So chylomicrons have the least proteins, and that's why they're the lowest in density, but they're the largest in size because they contain the highest percentage of triglycerides. And notice here they said chylomicrons contain the highest percentage of triglycerides. But if we go back to this slide, they tell us that elevations of plasma triglycerides usually are caused by increased VLDLs. So how come here they're talking about VLDLs having to do with increased plasma triglycerides, but here they say that chylomicrons are actually the ones that have the highest percentage of triglycerides? Well, think about it this way. Chylomicrons we obtain from our diet. Okay, so when we have a clinical, when we have some sort of disease that would increase our plasma triglycerides, this would have nothing to do with our diet, because our diet is the chylomicrons are based directly on diet, and they're not based on plasma triglycerides. But VLDLs are endogenously synthesized. So when we have high plasma triglycerides, these are directly correlated to increased VLDLs because these are de novo synthesis and not exogenous sources of lipids, as in chylomicrons. Okay, so VLDLs and LDLs are successively denser, having higher ratios of protein to lipid. So HDL, of course, stands for high density lipoprotein. LDL is low density. VLDL is very low density, and chylomicrons, they're just the lowest in density. Okay. So again, the slide is pretty low yield. There's not much that we have to know from it. All we have to know is that in the electrophoresis, the HDL travels the furthest, and that's because it's the most negative due to all the proteins, due to the high density of the proteins. Now, VLDL, although it's less dense than LDL, it has more proteins. Okay, so that's why it travels closer to the anode than LDLs. And then chylomicrons, of course, it's the least dense and it has the least proteins, so it stays close to the origin. So let's talk about apolipoproteins. So if you remember, we have our lipoproteins, and then we have everything that we talked about, our cholesterol, our phospholipids, and then our cholesterol esters and triglycerides in the center. And then on the outer surface, we have big proteins called apolipoproteins. Okay, apolipoproteins. So we have different kinds of apolipoproteins. What we want to remember is that apolipoproteins, as I mentioned previously, they can be transferred from one lipoprotein to another. For example, from HDL to LDL. except for the beta or the B apolipoproteins. Okay, so the B apolipoproteins are actually intrinsic constituents of their lipoprotein particles. That means they cannot be transferred. Okay, so when we say chylomicrons contain ApoB48, this ApoB48 belongs exclusively to chylomicrons. It cannot transfer to any other lipoproteins. Okay, and LDL and VLDL contain ApoB100. So just remember, chylomicrons, ApoB48, LDLs and VLDLs, ApoB100. Okay, and HDL contain the A apolipoproteins. Okay, so let's talk about the apoprotein functions. So first of all, they regulate lipid metabolizing enzymes in the blood. Now, what do you guys think they're talking about here when they say regulate lipid metabolizing enzymes in the blood? They're of course talking about ApoC2, because ApoC2 attached to the chylomicrons would actually activate the lipoprotein lipase on the endothelial surface that would cause the metabolizing enzymes to act on the triglycerides. Of course, we'll be going over this later on. Number 2, facilitate the transfer of lipids between lipoproteins. And number 3, bind to cell surface receptors for endocytosis or exchange of lipids with cells. So ApoB48 versus ApoB100. All we have to remember from the slide is that ApoB48 is basically just it's a part of ApoB100 that was basically that had a CAA codon that was post-transcriptionally changed into the stop codon UAA. That's why we have in the liver, it's ApoB100, as in LDLs and VLDLs, but in the intestine, because we have this CAA codon replaced with UAA, which is a stop codon, it's stopped prematurely, and of course it's in the intestine, and that's why chylomicrons are ApoB48, and VLDLs and LDLs are ApoB100. So lipoprotein lipase. Now this is a very, very high yield concept. A lot of the questions that you see from this lecture are related to lipoprotein lipase. So lipoprotein lipase is extracellular enzymes attached to capillary walls of adipose tissue, cardiac, and skeletal muscles, and they're not found in the liver. They act on chylomicrons mostly, and all VLDLs. Okay, so they're carried by, so they break triglycerides that are carried by chylomicrons and VLDLs. So the lipoprotein lipase, as I said previously, they're activated by the ApoC2 on the surface of the chylomicrons and inhibited by ApoC3. Okay, so if a chylomicron, if we happen to have high levels of chylomicrons in the blood, well, that tells us that there's an issue with ApoC100, because ApoC100 is used to activate the LPLs, and if we have a high concentration of chylomicrons in the blood, that tells us that the hydrolysis of these lipoproteins is not happening as efficiently as it should. So the regulation of lipoprotein lipase activity. In the fed state, LPL synthesis is increased in adipose, okay? And during fasting, they're increased in muscle. Why is that? Well, during the fed state, the adipose wants to store all of these triglycerides, and that's why we have an increased lipoprotein lipase activity, synthesis activity. But at the same time, when we're relaxing and we just had a meal and we're fed, our skeletal muscles don't rely on the triglycerides as their main source of fuel. So we have decreased LPL synthesis and activity during the fed state. However, when we're exercising or we're fasting, well, the muscles then rely exclusively and mostly on triglycerides and fatty acids as their main source of fuel. And that's why you have increased LPL synthesis in the muscle and decreased LPL synthesis in the adipocytes, because all of these triglycerides in the adipose tissue would then be redirected towards the muscle, all of these triglycerides. So we would no longer need to store all of these triglycerides in the adipose tissue. So as I mentioned previously about chylomicrons, they peak about 3 to 4 hours after a fatty meal, and that's why you would have to fast before you go in to get your blood cholesterol checked, because you would then get the transient hypercholesterolemia. Okay, so after a very high fat meal, chylomicrons cause serum to become turbid or milky. The term chyl refers to the milky fluid consisting of lymph and emulsified fats or free fatty acid, which is how the term chylomicron was derived. Of course, chylomicrons have a single molecule of ApoB48, and this is a intrinsic apolipoprotein, so it does not transfer to any other lipoproteins. And as I mentioned previously about the transfer of apolipoproteins, we have ApoE and the C apolipoproteins that are acquired in the blood by transfer from HDL. So lipoprotein lipase removes 80-90% of the triglycerides. And yeah, so everything we talked about previously is summarized in the slide. So let's go through everything we just went. Let's go through everything again. So we have in the intestinal cells, we have chylomicrons that are, of course, from exogenous sources of lipids from your diet. And then from HDL, you get added ApoC and ApoE, and then you get the chylomicrons that are circulating in the bloodstream. Then when they reach the endothelial cell, you have a lipoprotein lipase that then would be activated, of course, by the ApoC2 right here, and it would start metabolizing the triglycerides within the chylomicrons. Then we have the chylomicron remnants that would then go back to the liver and then would be endocytosed through the receptors. So they would undergo receptor-mediated endocytosis. Now let's talk about VLDLs. Now, as I mentioned, VLDLs are assembled in hepatocytes. So they are synthesized like de novo. So VLDL transport TG, VLDLs transport triglycerides containing fatty acids synthesized from excess glucose to adipose tissue and muscle. Okay, so we get liver to adipose and muscle, of course. And this is the function of VLDLs. VLDLs are released with a single molecule of ApoB100, small amounts of ApoE and C, ApoE and ApoC, and cholesterol esters. And they acquire these ApoEs and ApoCs from HDL, just like the chylomicrons, okay? Cholesterol ester transfer protein, CETP, helps VLDL acquire additional cholesterol ester from HDL. We'll be going over this soon. So like the chylomicrons, VLDLs is metabolized by lipoprotein lipase. And like chylomicrons, VLDLs transfer ApoC to HDL, but retain most of its ApoE. Now, this is pretty low yield. I don't think you would need to know a lot of these points. Smaller remnants appear initially as intermediate density lipoproteins, and then they eventually transform into LDLs. Okay, this requires the hydrolysis of excess triglyceride and phospholipid by hepatic lipase and transfer of excess apolipoproteins to HDL. Now, let's summarize this. VLDLs are released by the liver. They go to adipose tissue and muscles. They're then metabolized by the lipoprotein lipase. Then these remnants become IDLs and eventually LDLs. But before they become LDLs, these remnants would have to go back to the liver and then would have to go hydrolysis again through hepatic lipase. And then that's when they would become LDLs. And that's what this slide tells us. So from the liver cell, we get the VLDL. And then, of course, it acquires the ApoCs and ApoEs from HDL. It would then go down to the bloodstream. It would then start circulating in the bloodstream and would start getting attacked by lipoprotein lipase. So then these ApoCs, the triglycerides, and all of these things would go back to HDL, and we'd get a VLDL remnant. Now, the VLDL remnant either would go back to the liver cell and would become endocytosed, or it would go back to the liver and then would become processed again by the hepatic lipase to become LDL. Okay, and then this LDL again would go and drop off these cholesterol, and then of course like high LDLs would cause atherosclerosis and all of these things. So chylomicrons and VLDL transport triglycerides to adipose and to adipose tissue and muscles. The only difference is chylomicrons are endogenously made, or they store exogenously exogenous sources of lipids because of the diet, and VLDLs are storage of endogenously synthesized triglycerides and lipids. Okay, so that basically sums it up for plasma lipoproteins 1. Let's just go through a couple questions before we end off. A genetically based deficiency of which of the following will most likely result in decreased lipoprotein lipase activity? So which of the following, basically another way to word this question would be, which of the following is an activator of lipoprotein lipase? And the answer, of course, would have to be a ApoC2 on the chylomicron surface. So the next question is, a 23-year-old man is found to be incapable of producing chylomicrons. Which of the following is the most likely consequence of this disorder? So it's either fasting hyperglycemia, impaired absorption of dietary lipids, increased risk of hypertriglyceridemia, increased risk of lactic acidosis, increased serum urea nitrogen concentration. And of course, the answer is B, impaired absorption of dietary lipids. And finally, which of the following is involved in the transport of newly synthesized triglycerides from the liver to adipose tissue? So of course, as I mentioned, the keywords that you want to look for is newly synthesized, meaning de novo, or endogenous source, right? And the second keyword is from the liver. So of course, the answer to this would be VLDLs. And I'll be going over plasma lipoproteins in a second. Thank you very much.